We have a stellar lineup of speakers here for you today to bring you our webinar, COVID-19 Renal Susceptibility in Clinical Trials, which is presented by our NUGoKidney's Therapeutic Design and Development Core, or as we call it, Core B. Core B brings together the expertise and cutting edge techniques and services from a variety of different Northwestern cores to provide an efficient and translational conduit for access and expert guidance on studying kidney therapeutics development with the ultimate goal of clinical translation. I'm honored and excited to introduce our first presenter, Dr. Anand Srivastava. He is an assistant professor of medicine in our division of nephrology and hypertension, and he's a core member of the Center for Translational Metabolism and Health in our Institute for Public Health and Medicine here at Northwestern. His research focus is on patient-oriented research focused on the discovery and validation of novel, non-invasive blood, urine, and imaging biomarkers that will hopefully help identify high-risk patients with kidney diseases to help optimize the conduct of clinical trials and service tools for drug development. He has recently been working, he's been one of over 400 investigators working on the Stop COVID trial, which I'll let him tell you more about in his presentation today. And I will turn it over to Dr. Srivastava.
Great. Thank you very much for the kind introduction and for the opportunity to speak here today. Next slide. These are a list of my disclosures, none of which are pertinent and relevant for this talk. Next slide. Coronavirus disease 2019 or COVID-19 is caused by severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. And as of this morning is responsible for over 11.5 million infections worldwide with a resultant over 540,000 deaths. And in the United States, this is translated to nearly 3 million infected individuals, which we will likely surpass this week with a resultant over 130,000 deaths. And this is data here from the Johns Hopkins Coronavirus Resource Center that demonstrates that clearly we are still in the midst of a global pandemic. And in the orange line at the top is the United States, where we see a sharp increase in the confirmed number of new cases in mid-March, which has started to flatten out with now an uptick in the number of confirmed new cases, which has definitely caught the media attention as well in the public eye. Next slide. There are a number of complications of COVID-19, and typically at the top here, we see that the SARS-CoV-2 will attach and bind to the ACE2 receptor. I will say that 2 to 3 weeks ago, we had an outstanding webinar from Dr. Daniel Battier in our division, where really he gave an exhaustive overview of ACE2. And it's thought that after the binding and internalization of the virus, it can trigger a pretty profound inflammatory response that can result in sepsis and hypercoagulability. And you can develop other manifestations that are neurologic with rare reports of encephalitis, thromboembolic disease, hemodynamic shock and circulatory shock, meaning low blood pressure to keep, requiring medicines to keep the blood pressure up. You can have liver injury, cardiac manifestations like myocarditis, pericarditis, and what is typically reported as viral pneumonia and in its severest form can progress to an acute respiratory distress syndrome or ARDS. But today we'll focus more about the kidney manifestations, and what we typically will see are acute kidney injury in patients with COVID-19. And this is often detected by an increase in serum creatinine with a resultant decrease in the estimated glomerular filtration rate or kidney function. And so in individuals with a rising serum creatinine and a decreasing urine output, AKI in its severest form may lead to individuals requiring renal replacement therapy or RRT, which is the need for dialysis. Next slide. While we're well acquainted with taking care of patients with AKI, as are our colleagues in the hospital, this does not always reach media attention in the public eye. And I think that this puts it into perspective that during the midst of the early parts of the pandemic, just within 2 weeks of one another in the New York Times were articles that discussed that a key resource in patients with COVID-19 may not only just be ventilators, which was a lot of the demand that was required early on, but that dialysis machines may become a scarce resource in the midst of increasing confirmed number of cases. If you've not seen these articles, it's worth reading to see both a larger perspective and a patient's perspective as well. Next slide. But still at the beginning of the pandemic for us in the United States, we still actually did not know all of the kidney function abnormalities that we may see in hospitalized patients with COVID-19. This is one of the first reports, and it came from Wuhan in China, that demonstrated a cohort of over 700 individuals with COVID-19. And what they found was that the prevalence of acute kidney injury, or AKI, was around 5%. And I know a number of us in our community thought that this was likely an underestimate of acute kidney injury. What was a bit provocative was that they found that individuals hospitalized with COVID-19 also had proteinuria and hematuria present upon hospital admission. And the reason this is provocative is that typical sepsis-related syndromes that may lead to AKI often do not have protein and hematuria in the urine. And so this may suggest that there could be an alternate pattern of injury ongoing in the kidney that required further investigation. They also went on to see that individuals with higher levels of proteinuria and hematuria, as based on urine dipstick, were associated with an increased risk of in-hospital death, and that the presence of acute kidney injury was also associated with an increased risk of in-hospital death. Next slide. Now, more recently, we've had data from the United States, and this is actually over 5,000 patients at 13 hospitals in New York, both academic and community. And they have uncovered that the prevalence of acute kidney injury in patients with COVID-19 is probably closer to 37%. And when you look at different staging of AKI, the severity, that individuals with stage 2 was probably around 22%. That's marked by a doubling of your serum creatinine. And that stage 3, which is tripling of your serum creatinine or the need for dialysis, was around 30%. They also identified certain independent risk factors for acute kidney injury, namely older age, being of Black race, hypertension, diabetes, cardiovascular disease, the need for vasopressor use in individuals who are critically ill, and the need for mechanical ventilation in individuals who are critically ill. And in the individuals with acute kidney injury, still in that study, about 40% of them were still admitted to the hospital, but they saw that the mortality rate in these individuals was 35%. Next slide. Interestingly, in individuals who had urine studies that were collected at the time of acute kidney injury, which is about 30% of the individuals with AKI, they also were able to identify that over 30% of the individuals had higher grades of hematuria and proteinuria upon their presentation with acute kidney injury. And on the right, I show a table here that shows that in AKI of its severest form, those that need dialysis, highlighted in red, that individuals that were mechanically ventilated, so critically individuals, they actually had a 23% prevalence of the need for dialysis. When you compare that to individuals who were not mechanically ventilated, that was almost 100 times greater prevalence for the need for dialysis. Next slide. So I think that the urine findings actually stimulated a significant amount of discussion in our field about, is the pattern of injury different than one would expect? So typically somebody that has a sepsis-related acute kidney injury, again, as I mentioned, the urine would be relatively bland and you would not see blood in urine. But could there be something else? And if you were to look at a biopsy of those individuals of the kidney, and you would probably see tubular injury, meaning that the tubules themselves in the kidney were dilated and you would see sloughing of cells, or they could become necrotic. And so this was actually a study in Wuhan of 26 autopsies in patients with COVID-19 that died of respiratory failure. Now, only 9 of the 26 actually had a clinical signs of acute kidney injury. And when they looked under light microscopy, they actually saw, yes, that there was acute tubular injury marked by dilation of the tubules and sloughing of the cells, but they also saw that there were red blood cell aggregates in the peritubular capillaries. I think what was of significant interest and sparked some degree of controversy was under electron microscopy, that the red arrow actually points to certain parts of the slide there that looks like virions from coronavirus, which would suggest that maybe there was direct viral invasion of the kidney from the virus. This has been refuted a bit in the literature, though, that in fact, in individuals that have significant tubular and podocyte injury of the glomerulus, that you can actually have a multivesicular body form, and that some of the protein that outer part of the vesicle, clarithrine, can actually look like electrodense and give you almost a corona-like halo. So it is still not clear whether or not there is direct viral invasion of the kidney or not. Next slide. What we have seen is a bit of a dramatic presentation of acute kidney injury as well. So this is a case series of 6 individuals with COVID-19 who had a very dramatic presentation in the sense that their creatinine was severely elevated and they had massive levels of proteinuria. And so a normal amount of proteinuria per day in someone without kidney disease or kidney dysfunction would be under 200 milligrams per day. And these individuals on the median had 11.5 grams. So all these individuals underwent a kidney biopsy, and in panel A, they saw a collapsing of the glomerular tuft. This is a very classic finding for a collapsing glomerulopathy. And in panels B and C, you can see that they did in fact see tubular injury, as one would expect, with some degree of interstitial inflammation. And in panel D on electron microscopy, they saw extensive foot process effacement of the podocytes, which would explain the massive amounts of proteinuria. Now, this entity of collapsing glomerulopathy, it seems to be slightly more predominant in individuals who are African American. And so the investigators went on to actually genotype them for APOL1 high-risk variants. Now, it is known in our space that high-risk variants of APOL1 are more common and increased in patients of African ancestry and may provide protection against the trypanosomal parasitic disease in Africa. But we also have seen that individuals with high-risk variants are increased for the development of chronic kidney disease and of kidney disease progression over time. And we've also seen that individuals who have high-risk variants are more susceptible to developing collapsing glomerulopathy. Oftentimes, this is triggered by another viral infection, and what has been most notable is in HIV. And so we have seen something like this before, where individuals who have high-risk variants may get a viral infection, and it could trigger a collapsing glomerulopathy and a very dramatic presentation of acute kidney injury. Now, this is not to say that all individuals with protein and blood in their urine may have some intermediate phenotype of this, but rather that there can be a very flagrant phenotype in patients with COVID-19, and additional research is needed in order to identify particularly vulnerable populations. Next slide. So I've shared with you some more regional data both in Wuhan, but also in the United States about patients who develop acute kidney injury. But particularly susceptible populations may be those that are critically ill. And we wanted more nationwide representative data. So I've been fortunate to be a site PI here at Northwestern for the Stop COVID study, which is a multi-center observational cohort study of critically ill patients with COVID-19 admitted to intensive care units across the United States. And today, this has now included over 5,000 patients at 67 hospitals with over 400 study investigators. What's different about this cohort is that actually we have a fair amount of granular level of data, as all the data elements were extracted manually from the patient's chart. So there's over 800 data elements per patient. Next slide. There are really 3 main goals of Stop COVID, and that is 1, to investigate the clinical features and outcomes of critically ill COVID-19 patients, namely death and acute organ injury like acute kidney injury, acute respiratory distress syndrome, and thromboembolism. 2, to perform causal inference analyses of the effects of treatments on outcomes, and to assess inter-hospital variation in treatments and outcomes. And I'm going to share some data on bullet number 1 with you. Next slide. When we look at the timing of organ injury, we see that from the time of symptom onset, that acute kidney injury seems to occur at about 10 days after a symptom onset. And since these are all critically ill individuals, that this seems to occur about 3 to 4 days after ICU admission. Next slide. When you look at the prognosis of individuals who are critically ill with COVID-19, that the overall 28-day mortality in this cohort was around 35%. This is similar, slightly higher than what was reported in the Italian perspective in JAMA not too long ago. When you look at individuals who have acute kidney injury, and in this analysis defined as a doubling of serum creatinine or worse, that the incidence appears to be around 43%, and that their 28-day mortality can be around almost 55%. And once you compound additional multi-organ dysfunction, you start seeing higher rates of mortality. Next slide. So what was lacking in some of the other studies is more data about underlying kidney function, because they were not able to obtain some of that data at baseline. And so this is actually looking at the sequential organ failure assessment renal score. So looking at their actual kidney function marked by serum creatinine and their urine output upon ICU admission. So individuals who had a score of 0 had relatively preserved kidney function with good urine output, and with worsening score, you had worsening kidney function with declining urine output. And what we see is that decreased kidney function is independently associated with death, and almost in a stepwise relationship. And that with individuals with a score of 3 or 4, they're at over 2-fold increased odds of 28-day mortality. This model also included other demographic and risk factors for mortality in the ICU, and in red are other independent risk factors for 28-day mortality. Next slide. So to summarize, AKI commonly occurs in COVID-19. It's associated with an increased risk of death. Hematuria and proteinuria often are present in patients with COVID-19, with or without acute kidney injury, which may suggest possibly that there's an alternate pattern of injury than your traditional acute tubular injury that we may see. Decreased kidney function increases the risk of AKI-RRT and death in patients with COVID-19. So in addition to other risk factors that have been demonstrated, higher body mass index, worse hypoxemia, and higher D-dimer appear to be independent risk factors for AKI in its severest form, those that need dialysis. Dialysis dependence still remains high upon hospital discharge, and additional studies are still required to identify predictors of kidney function recovery. And lastly, novel strategies are needed to provide earlier detection of AKI, and therapies are still needed to improve the outcomes in patients with COVID-19 who experience AKI. Next slide. I'd like to take a moment just to acknowledge our collaborators here at Northwestern University, but also the site PIs and the coordinating site at the Brigham and Women's Hospital for Stop COVID, that is Shruti Gupta and David Leaf, and of course the other Stop COVID investigators. There are very many of them. It's a real pleasure to work with them. And also for my funding support through the NIDDK and through NUCATS, and for NUGoKidney for their core resource support. Thank you very much.
Excellent. Well, thank you very much, Dr. Srivastava, for that incredible presentation. We have a few questions already coming through our Q&A, which we'll save to the end. But if you have any others, again, as we go through the presentation, feel free to put them in that Q&A box there. Next up, we have Dr. Michael Ison, and he is the professor of medicine in the Division of Infectious Diseases and also professor of surgery in the Division of Transplant Surgery here at Northwestern. And he's also the chair of the Adaptive COVID-19 clinical trial at the National Institute of Allergy and Infectious Diseases Division of Microbiology and Infectious Diseases. His research studies focus on areas including respiratory viral infections and organ transplant recipients, and he's here with us today to share his insights from data on clinical care from Northwestern Medicine about the evolving epidemiology of COVID-19 and its impact on patient outcomes. Dr. Ison, thank you for joining us, and I'll let you take it away.
Great. Thank you so much for letting me chat with you today. Next slide. Here are my disclosures. Next slide. So I just wanted to highlight some resources that you may or may not be aware of that I'm involved with. The first is a large effort led by Chad Achenbach that is establishing a data mart and treatment cohort assessment program through the EDW. This has been a great opportunity to leverage some work that I had ongoing for a long time into COVID-19. We've gotten support from Gilead and other companies with the help of the Center for Health Information Partnerships to pay for a lot of the work that's been ongoing. Next slide. So really this was brought up to answer a couple of different questions. What are the evolving epidemiologies of COVID-19? Who's being admitted for and at risk for poor outcomes? And how are we managing COVID-19 in the hospital? Specifically, where we've started directing our attention is we have had a number of treatments: convalescent plasma, tocilizumab, hydroxychloroquine and others that haven't been part of clinical studies, but a relatively large number of patients have received. Sometimes there's been combinations of various interventions. And the way that we're planning to look at this is to develop matched controls for each of these patients that receive these various therapies to look at their outcomes retrospectively. Next slide. But in a nutshell, how we've collected this data is it's a clinical cohort study of all adults diagnosed with COVID-19. This is either lab confirmed ICD-10 coding, or if they had an infection prevention flag. The reason for these various indications is that throughout the course, particularly early on, when we weren't having sufficient testing, some patients may have had a diagnosis that wasn't laboratory confirmed. We've pulled a fair amount of data directly from the EDW, but we've done individualized case reviews of the medical record to pull things such as symptoms, imaging assessments, and exposures that may not be able to easily pull from the record. We've looked at outcomes, including where they are on the ordinal scale that's been defined by a number of groups, including the World Health Organization, as well as durations of intubation and some composite endpoints. Next slide. So looking at the total number, this is from the NMI webpage. We continue to have on over time about 8.5% positivity rate in our testing. Recently, that's dipped down because of declining rates. Next slide. Overall, we've had a little over 3,600 individuals admitted to the various NM hospitals, about 1,300 at Northwestern. Thankfully, we've seen a significant decline in the number of hospitalizations over time, particularly in the ICU. Next slide. Looking at data from the EDW, when we took this snapshot, which is a few weeks old, we had about 5,000 patients. These include hospitalized and outpatients. Hospitalized accounts for about 40% of our overall data set. The average age was 51, 23% were over 65. Again, highlighting that it's not just older adults that are getting sick. About 18% were Black race, 34% Hispanic. We're going through because there were a lot of missings. Prior smoking was present in about 20%, and about a third of the patients lived here in Chicago. Next slide. In terms of epidemiology, this again was through the end of May. Shown here, you can see we had pretty stable rates throughout much of April and May. Thankfully, that's begun coming down nicely. Next slide. And hospitalizations really followed a similar trend with a clear decline in hospitalizations. Next slide. This is same is true for ICUs. Unlike hospitalizations, these didn't taper off quite as much as quickly, in part because around the middle of May, we reduced the number of admissions from outside the hospital, but increased the number of people that were being admitted as transfers in from other facilities. Next slide. And then this is looking at the some interesting findings that we noted in terms of ethnicity. So we had a reduction of patients that were non-Hispanic over time, starting at the end of April. At around that same time is when we started seeing an uptick in the Hispanic population, particularly in some of our partner hospitals within the NM system. Next slide. In terms of outcomes, about 40%, as I mentioned, of patients were hospitalized. We have an overall mortality of about 3%. This includes outpatients and inpatients. About little over a quarter of patients required ICU level care, 17% of the hospitalized patients required intubation, and of those that were hospitalized, about 85% were discharged alive at the time of this analysis. Case fatality rate, again, among hospitalized patients was about 7%, among ICU patients about 15%, and intubated patients about 21%. And it's important to recognize all of these rates are a little bit lower than some of the reports coming out of New York and cities in the Northeast, and there's a number of reasons for this potentially. The average duration of hospital stay was around 8 days for those hospitalized, a little longer for those that ended up dying or required intubation. Next slide. So just briefly, since this is a kidney population, on the solid organ transplant group, this was an effort led by Sejal Tana and 2 of our fellows. We had about 35 patients as of about 2 months ago when we submitted this for publication. We're now up to, I believe, 74 patients, the majority of which are kidney transplant patients, as I'll show you on the next slide. Interestingly, there was a higher proportion of patients presenting with diarrhea, particularly early on in their clinical course, and things such as fever were far less likely, particularly in patients with milder disease, those that only required the floor or outpatient management. Next slide. I'll skip this and move on to the next slide. So much like the rest of our patient population, there were very few co-infections. We've actually been a little bit surprised, given the significant inflammatory component of this disease, how few of our patients have developed typical OI complications such as CMV. Thromboembolic events have been relatively rare. Thankfully, we've had very few patients die. I believe we're up to 4 patients at this point, still with a mortality overall of about 4 to 5%. Those that were admitted to the hospital stayed for about 9 days, and renal replacement therapy, thankfully, was not common among this patient population. Next slide. Now, one thing that we found to be very interesting are 2 features. One related to race, so in the green bar is the overall transplant population over the last 10 years here at Northwestern. The blue bar are those patients that were diagnosed with COVID-19. And you can see there's an overrepresentation of African Americans and an underrepresentation in Caucasians. Additionally, all of our patients that required ICU level care were non-Caucasians in our population here, again, highlighting that this risk factor may be expanded in the transplant population. The other thing that we found is that there was a slight overabundance of kidney transplant patients and an underrepresentation of our liver transplant population. Additionally, there was skewing to more patients that were late post-transplant, and this was greater than the proportion of our overall population. So in my mind, this suggests that patients further out really do not probably take their transplant status as seriously as they get later out from the transplant. Next slide. One feature that we've noted is that these patients have very prolonged shedding. At the time that we looked at this about a month ago, the average duration of shedding was somewhere around 40 days. We now have a number of patients that are well beyond 60 days of shedding, and some of these patients have relatively low CT values, meaning that there's a fair amount of virus present. Next slide. The other critical feature is the impact that COVID-19 had on overall transplant volume. On the left is the national data for the whole Great Lakes region, and then to the right is our Northwestern specific data. And blue is kidney transplant population, and much like the rest of the country, we had a precipitous decline in the number of kidney transplants performed over the couple of months that elective surgeries were discontinued. This is starting to bounce back, thankfully, and is something that the team is working very carefully, although there's some new challenges, especially for some of our living donors. As a kind of shout out to our lung transplant team in the gray line, they were the one that really actually increased volume and have been doing some really interesting things, such as the double lung transplant in a patient recovering from COVID. Next slide. Now talking about some potential resources that we have available for us. So we have the Northwestern COVID-19 Biobank. This is a prospective study of patients that are hospitalized with PCR-confirmed SARS-CoV-2. They have nasal swabs collected for every 4 days until discharge or through day 30. They have blood collected at day 1, 8, and 16, and urine collected at day 1, 8, and 16. We just started additional testing for the PAI-1 levels, which will get our first signal back. We have a number of planned analyses, including sequencing of the sample, and I'll show you some of that data on the next slide. Assessments of viral loads, and I'll show you that as well. And then planned, although we haven't started it yet, proteomic or transcriptomic profiles. And I really want to give a big shout out to the collaborators on this, Judd Hultquist, Ramon Lorenzo Redondo, and Egan Ozer, as well as Lacey, who works in Judd's lab, who's really done a huge amount of the work here, and our two fellows that just graduated, Hannah and Scott, that without them, we would have been dead in the water. I think, interestingly, looking at the patients, we also have a number of patients that have received a range of therapies. Green are patients that have received remdesivir. Blue were not enrolled in any trials, but some of them got tocilizumab. And then the pink are patients that got sarilumab therapy. Next slide. So one of the things that we did is that we looked at viral loads, and this is CT values. This is increasing over time, which is not too unexpected. Interestingly, we have found a couple of mutations that have emerged on therapy, suggesting that some of the antiviral agents may in fact be inducing mutations, and we're doing further studies ongoing to understand the location of those and if they correlate with potential resistance. Next slide. This program is part of a larger effort channeling through Judd Hultquist's lab that's pulling in a number of viral sequences from programs across the city. We also are recovering residual specimens on all patients that have PCR-based testing, and then this is being studied for sequencing with analysis by Ramon Redondo's group. Next slide. And with that, we have a paper that is undergoing review in which we were able to describe the makeup of the viruses infecting the majority of our patients. Most of the viruses link up to either clade 1 or clade 2. Clade 1 is the virus that is expanding most significantly. This appears to be a virus that was introduced into the New York City area and spread throughout the country, likely coming from Europe to New York and then beyond. This is nationally and here in Chicago increasing quickly. This is the one that has significant interest because of some unique mutations present in the genome. Clade 3 is the virus that emerged, came from China into the Washington state area, and then was transmitted beyond that. Interestingly, though, we also identified clade 2. This virus appears to be relatively unique to the Chicagoland area. There have been some exports from the Chicagoland area, but likely developed here in Chicago, and may be a precursor of one of our early introductions. Interestingly, this virus is associated with slightly increased viral replication as well. Next slide. The last resource that I'm leading is the Northwestern COVID-19 Convalescent Biobank. This is funded through the Lurie Cancer Center. I forgot to mention the prospective biobank was funded through a NUCATS grant. So in this biobank, we're collecting samples from patients that have recovered from COVID-19. These include 3 groups of patients that we've identified and were never hospitalized at Northwestern. 2, we have identified patients that are part of our prospective study in the hospitalized setting that are discharged and coming back in. And then 3, working with the group with Beth McNally, those that are from the community that are getting tested that have high titers. There are other groups that have reached out to us that we are hoping to get engaged. There's also an effort that we're leading to look at whole genome sequencing of patients either in these studies or others. And we just submitted the amendment to this biobank to do a collection of relevant tissue and blood to be able to do whole genome sequencing to look at the array of clinical expression that we're seeing in these patients, but also immunologic responses. Next slide. So it's really been an honor to be part of this group, and really the person that's been doing a lot of the science behind this is Huiping Liu, who's been critical in developing a group of experts that's using much of the blood that we're collecting from the convalescent plasma to basically be processed and aliquoted for future use. We're doing flow cytometry. They're also looking at other unique components, and one of the things that we've found from this data so far is that the number of B-cell clones is relatively low compared to other infections in people that are recovering from COVID-19. We've identified a few unique antibodies that are being shopped around as potential therapeutic options for these patients. There's a number of other studies that are planned that haven't been completed, looking at proteomics and exosomes that are ongoing. Next slide. So it was an honor to chat with you a little bit about some of the things that we have ongoing, and I'll turn it back over to Ashley to move us on to the next part.
Thank you very much, Dr. Ison, for that fabulous and very comprehensive presentation. We have a little bit of time left in the time that we've allotted for your presentation. Would you mind if I threw you a question now?
Go for it.
All right. So we have a question from our Q&A. Do you think we will see another surge in patients? And if so, are we better prepared to deal with it? And will we be able to reduce ICU and hospital admissions?
Yeah, lots of questions there. Number one, I can guarantee we will have another wave of disease, whether it is unfortunately a bump in our current numbers or truly after things are calm and then a true second wave is unclear. We've done a really good job in trying to limit travel from high-risk areas back here. But I think as all of us have probably seen, I would estimate that only about 1/3 to 40% of people in public are using masks. And so I think we've learned when we don't focus on public health measures that we will likely see what they're seeing in the South and West with the emergence of the virus coming back. Will we see differences in the number of admissions and ICU admissions? Well, I think we've learned a lot. You know, I didn't talk a lot about some of the trials that are ongoing, but we have some therapies, dexamethasone and remdesivir, that are potential therapies for these patients. There's some really exciting things that are coming along the pike, including an oral antiviral that a lot of us are very interested in, that may allow us to reduce the risk of progression. Additionally, I think we've learned a lot about how to test and manage these patients, and hopefully we'll have better outcomes from this. I think the biggest thing that we've learned is that by not overwhelming the healthcare system, you can have better outcomes. And this really doesn't depend on us. It depends on the community. And one of my big fears, for example, is in Florida, Texas, and the West, is as these hospitals become overwhelmed, they may not be able to give the same level of care as we were able to give our patients, and may have worse outcomes.
All right. Well, thank you so much again for joining us. We have a few other questions for you, so we'll get to those at the end.
Sounds good.
All right, thanks again. So next up, we have Dr. James Mathew, and he is a professor in the Division of Organ Transplantation and Microbiology and Immunology here at Northwestern Steinberg School of Medicine. He's also the director of Northwestern's Immune Monitoring Corps, and he's an NUGoKidney Core B navigator. His research for the past 25 years has been in the areas of immune responsiveness and tolerance induction in clinical organ transplantation. And he's with us here today to share what we know so far about the immune response to COVID-19 and also some of the potential techniques that could be applied in studying this further. Dr. Mathew, thanks for joining us.
Thank you, Ashley, for the introduction. And for the next 5 minutes, I will talk to you about the immune responses to SARS-CoV-2 and some of the assays that we have developed at the Immune Monitoring Core of the Comprehensive Transplant Center. Next slide, please. Last week, I performed a literature search on cellular immune responses to COVID-19 and found just 5 papers. And this particular one is a review paper which was published last month in Immunity. And it showed that there is a decrease in the peripheral blood T cells with disease severity and inflammation. And that the residual cells that are there are actually activated cells, some of which display exhausted phenotype as well as regulatory phenotype. And most of these cells secrete inflammatory cytokines. Of course, this needs to be confirmed in the future. Next slide, please. This slide shows another study from China, and they performed ELISpot assays in COVID-19 patients as well as normals against the nuclear protein, M protein, and the S protein, and found that the patients had different variable amount of response to the COVID antigens, but not in the healthy subjects. Also, they found that there is a correlation between neutralizing antibodies and the number of NP-specific T cells. Next slide, please. Oops. This slide, this is another study, and this is actually the most comprehensive study, and it is from the Loyola Institute of Immunology. And they assayed COVID patients, convalescent patients, as well as unexposed individuals, and found that nearly 100% of the patients, COVID patients were positive for CD8 and CD4 responses to the virus. And this is not only to the spike protein, but also to the M and N proteins. And also normal subjects, unexposed subjects have reactivity to the COVID antigens, and possibly due to common cross-reactive antigens from the other coronaviruses. Next slide, please. Because of the paucity of cellular immune responses studies, and because we are interested to study our transplant patients, we have developed a number of assays in the Immune Monitoring Core. And since we are part of the NUGoKidney program, and in the Core B, we are planning to provide these assay services if somebody in the group wants. Next slide, please. First, we developed a 14-color flow cytometric analysis panel for analyzing the immunophenotyping the patients for basic cell subsets, as well as for B subsets and Bregs, T cell subsets and Tregs, and also the first tube for the fate of the T cell subsets in terms of energy, exhaustion, and senescence. Next slide, please. We also developed a proliferative assay to using thymidine incorporation to the spike protein, membrane protein, and the nuclear protein in normal subjects. And depending on the subject, the assay, the response is marginal or a very little response is there so far. We are also developing a CFSC dilution-based proliferation assay to SARS-CoV-2 peptides. Next slide, please. Now, most of you must have heard about single-cell RNA analysis. A company called Isoplexis has got an assay for functional proteomics in terms of 32 cytokine estimation. In the panel A, that is the assay, how the assay is performed. What we do is, we are adapting the Isoplexis assay to COVID-19. What we do is we take the PBMC from the patients, or from right now normals, and then stimulate with the viral peptides, and after 36 hours, purify the CD4 and CD8 cells and load onto the chips which are produced by the company. Now, the beauty of the chip is that there are more than 3,000 channels as shown in panel B that are into which there is only one cell is being injected. If there are more than one cell in any particular lane, that lane is not counted. Now, across the lanes, there are 32 capture antibodies for specific cytokines are present. And if a single cell secretes the antibody, and that will be captured by the capture antibody, and then in the back again in the panel A, you will put back in the we put the chip in the instrument and run it overnight, and then analyze the data. Now, coming to panel D, depending on the data or the presence of different cytokines, whether effector, stimulatory, chemoattractive, regulatory, or inflammatory, we can characterize the patient or the person and person's potential response. So this is a powerful technology. Next slide, please. So we are thinking of providing this service to others. Also, you are welcome to contact me. And next, please. And I have to disclose that we have not started working with COVID patients yet. We have been working with only normal subjects. Thank you.
All right, thank you, Dr. Mathew. Next up, we have Dr. Evan Scott. He is an associate professor of biomedical engineering and the director of PhD admissions in Northwestern's McCormick School of Engineering. And he is an expert in polymer chemistry, nanomaterials, biophysics, and immunotherapy. And his research aims to regulate the immune system via rational design of nanomaterial-based delivery systems that target key inflammatory cell populations in various organizations. And we're honored to have him as part of NUGoKidney's Core B as a core director, where he's helping apply some of the techniques that he'll tell you about shortly to the kidney. Take it away, Dr. Scott.
Okay, thank you for that very kind introduction. You can hear me okay? Yeah, so I just very quickly, in these last few minutes, want to go over how Core B can potentially help others in the Northwestern community as well as beyond with their either kidney or COVID-19 related research. So essentially, Dr. Srivastava gave a really nice introduction and description of the relationship between COVID-19 and kidney disease. And so there are a lot of open areas of research currently, whether it's looking at direct targeting of the virus to podocytes and various cell types within the kidney, or the effect of cytokine storm, or comorbidities, or hypertension that can be related as well, resulting in heightened infection and death rates for patients. There's a lot of research ongoing in terms of COVID-19 in the kidney. Next slide, please. And we offer services with nanomaterials and nanocarrier dependent delivery. So if you have a therapeutic target, you have drugs that you'd like to investigate, you have specific cell types that you're interested in investigating, we can design nanocarriers to transport immunostimulants, various drugs and cargoes to those locations. And you can do that by decorating the surfaces of the particles with different targeting ligands like antibodies and peptides, for example. We can enhance targeting specific organs and tissues. We can target the kidney. We can avoid the kidney if necessary. Say your issue is you have kidney toxicity, we can design systems that can actually avoid uptake in the kidney as well. Next slide. And so nanomaterials and nano delivery is a strength at Northwestern. And so we're taking advantage of several different PIs on campus. And so shown here is work from Chad Merkin's group with spherical nucleic acids, allowing us to have access to a wide range of different types of chemistries and materials for imaging and delivery of nucleic acids and proteins as well. Next slide. As well as my group, who makes a variety of different types of polymer-based materials. Just quickly in the top right, we have nanomaterials that can result in slow release, like scaffold, slow release of drugs. We can have small micelles that can target specific cell types. We can have vesicular structures that can have an aqueous core to hold hydrophilic drugs or water-soluble drugs, as well as hydrophobic payloads at the same time. And we have some unique structures like this bicontinuous nanosphere structure. I'm not going to go into too much detail with, as well as other nanogel platforms, just to give a wide range of different capabilities to researchers at Northwestern and beyond. Next slide, please. And so just some of the general capabilities of these systems, as well as nanomaterials in general. Our systems are all completely scalable, so you can make large amounts of them for targeting in, say, large animal models or higher order studies. We can load a variety of different molecules, essentially any molecule of interest you like to load. We can load enzymes, peptides, proteins, DNA, RNA for controlled targeted delivery. We can do multimodal imaging as well, so you can see where your drug and payloads are going when you deliver them at the organ level as well as at the cell level. We design our materials for intracellular delivery, so we can enhance intracellular stimulation pathways, certain biochemical pathways. All of our materials have been demonstrated to be non-toxic, both in mice as well as in non-human primates. And one of the key advantages, as I mentioned, for nano delivery is the avoidance of side effects, whether it could be kidney toxicity or toxicity elsewhere. We can design the materials to target those select populations of interest and avoid toxicity elsewhere. Next slide. And just want to quickly point out that with everyone working with COVID-19 and vaccines in general, nanomaterials and nanocarriers are a key component of most vaccine formulations. And so our group in particular has a lot of background in developing vaccines, and this is just one example of a collaboration we have that's ongoing with Boston Children's Hospital. They have a precision vaccines program where we develop nanomaterials and delivery systems for them. This is just one publication we had with them a few years ago, and it demonstrates we can load a variety of different antigens and adjuvants into our materials. They're very stable, and through the precision vaccines program, we have access to a lot of different antigen and adjuvants that are difficult to obtain. Next slide, please. And so with that, I'd be happy to take any questions you might have. I just want to point out that if you'd like to discuss how to incorporate nanotechnology into your program or to your research, please feel free to contact me directly. I've also helped others with writing grants as well as proposals that incorporate the nano component into their proposal. So if you're not so familiar with how the chemistry or everything could incorporate into your proposal, I'd be very happy to help with that and even provide text if necessary to assist with your grant applications. And with that, I'll turn it back over to Ashley.
Oh, well, thank you, Dr. Scott. And if all of our presenters will rejoin us via video, I think we have time for maybe one question. This one's for Dr. Srivastava. Do you know if there is any change in the percent of AKI patients in new hot zones like Florida and Texas, reflecting a younger population?
Thanks for that question. It's very important. Unfortunately, no, we don't know that information yet. And I think with what we're seeing now with the more recent uptick in the number of new confirmed cases, it's still going to be some more time before we start to see what some of those additional risk factors or if there are demographic changes in patients with COVID-19 who develop acute kidney injury.
And then one more question for you. Is there anything known about demographic data of patients with proteinuria in China and or Europe where the prevalence is high risk of APOL1 genotypes is predicted to be lower than in the US?
That's a great question. And again, I think that in this situation, to my knowledge, we've not seen it broken down by proteinuria or not. And I think in part it's because often proteinuria data is not available in a lot of patients, or it's not being looked for, especially with all this retrospective type of data. But traditionally, the risk factors have been similar in Asian countries and in Europe. Obviously, in the United States, we have a little bit more with regards to body mass index that we can evaluate, but we still do not know if there are significant differences by proteinuria or not within different patient populations.
Great, thank you. I want to be mindful of everyone's time because I know, especially now, there's a lot of demands and people are getting pulled in all different directions, it seems. So I think we'll stop there. If there's any questions we didn't get to, we'll try to answer them offline and in our follow-up email to the group. I want to thank all of our speakers again here today for joining us and for all of you for taking the time to join us and giving us your attention. If you'd like to learn more about future webinars, you can go to our website, nephrohub.org. We're very active on Twitter, so you can also find it there. We're at NU_nephrology. You can also sign up for our email list. If you're interested in scheduling a consultation with either Dr. Scott or Dr. Mathew, you can also find out how to do that on nephrohub.org. You can just click the Get Started button that you see there on the slide, and then it'll guide you from there. So, like I said, keep an eye out for the recording and then a survey to tell us how we did. And thank you again, and we hope to see you next time. Stay safe, stay healthy, and stay encouraged. Thanks again. Bye-bye.